Welcome all, and thank you for joining today's Adabri webinar. My name is Matt Holdstock, and I work in Extension at the Adabri. In this session, we'll venture into the vineyard, and we will have a webinar titled Status of Fungicide Resistant in Australian Vineyards and Developing Strategies for Monitoring. In the spirit of reconciliation, the Adabri acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I'd also like to acknowledge Wine Australia for providing funding and support for the webinar program via the Adabri's Communication and Extension project. Now, before we jump in and make a start, a couple of quick reminders to anyone who is new to the Adabri webinar program. If you'd like to provide a comment or ask a question throughout, please click on the Q&A button on the Zoom toolbar, type in your question and click to send it through. And you can do this throughout the webinar at any time. We'll also hold a Q&A session at the end of both presentations, but feel free to send in your questions at any stage. And a reminder also that the session is being recorded and a link will be sent through to you later via the Adabri's YouTube channel. For anyone who's just joined, welcome. Today's webinar topic is Status of Fungicide Resistance in Australian Vineyards and Developing Strategies for Monitoring. And we will have two presenters today. The first presenter today is Dr. Ismail from the South Australian Research and Development Institute in Adelaide. Dr. Ismail has a Bachelor of Agricultural Science in Plant Protection and a PhD in Plant Pathology from the University of Adelaide. Ismail has 20 years of experience in plant pathology at various organisations and is currently the principal investigator on fungicide resistance research based at SARDI in the Horticulture Pathology Group here in Adelaide. So Ismail, if you're ready to make a start, I'll just get you to share your screen.
All right. Can you see my screen now?
Great. Thank you.
Excellent. Thank you, Matt, for inviting me for this webinar. And thanks for the people who are attending in this webinar. Today, I'm going to talk about status of fungicide resistance in Australian vineyard and developing a new strategy for monitoring. This research is funded by CRC SAFE, Wine Australia, and it's cooperation between SARDI and Curtin University and AWRI. So let's get started. For the beginning, I just want to apologize for people who already seen this slide before, but I just like to set up the background of what's the fungicide resistance. So the fungicide sale value has jumped from just below $280 million in 2019 to almost about above $500 million in 2023. So we're using a lot of fungicide in Australia. This part of this fungicide used to control powdery mildew, downy mildew, and Botrytis because they are the most significant disease in vineyard. These diseases are powdery mildew costs $76 million, downy mildew $63 million, and Botrytis $52 million. And because we need to control this, unfortunately, application for fungicide may lead to fungicide resistance. So what is the fungicide resistance? I just want to move this little screen from my face. Yeah. So what is the fungicide resistance? It's a pathogen changes from sensitive to a fungicide to less sensitive, or we call it decreased sensitivity or reduced sensitivity to, or insensitive, will become resistant. There is a few mechanisms involved in this process. Inside the cell of the pathogen, there is like a mechanism called detoxification, which means the cell degrade the chemical here, becomes ineffective, or they can mutate the target, and the chemical is not binding or overexpress that target a lot, so it's not enough chemical to kill the cell. Or they create a new target for the chemical, and then it's become ineffective. And the most common one, it's they pump out all the chemical outside the cell. So that's the most 5 common resistance mechanism. There's so many factors influencing the resistance. One of them, it's a DNA mutation in a fungal cell that's happened naturally. We don't have control on that. And then we have the factor of fungicide, what type of fungicide we apply. If we apply multiple mode of action, that's most likely to reduce the resistance. If you apply single mode of action, that's most likely to increase the resistance. Sorry. The fungal biology as well. So, for example, if you have powdery mildew can finish their life cycle in 5 to 7 days. And that's, imagine if you have resistant population, it can build up very quickly in the vineyard. There is the other factors, fungicide application, like the rate. If you underdose, the rate will affect that. The coverage, the number of spray you apply, and timing when you apply these fungicide will also influencing the fungicide resistance. This table shows how we assess the risk of fungicide resistance. As you can see in the left side of the table, that the fungicide class risk, it's high. Some of them high risk when they have one mode of action, and medium risk, and low risk when you have multi-mode of action. And the other side of the table, you have the pathogen, where you have a low risk of developing resistance, medium risk, and high risk. Unfortunately, we have our pathogen sitting between high for Botrytis and medium for Erysiphe, necator for Powdery, and Plasmopara, viticola for downy. So how does that work? If you have a high risk pathogen developing resistance, and you apply single mode of action, you multiply this number by this number, you get a high combined agronomic risk. And if you apply multi-site of action, you get less likely to develop resistance. And that's how the guideline for using to assess the resistance. There's two types of resistance here. We have qualitative resistance, which is in QI. Imagine this is sensitive population. It's normally that's what's look like. So stay always sensitive. However, in qualitative resistance for QI, it's one jump from sensitive to resistance. That's you only need one single mutation. That's normally cross resistance between all members of the group and generally stable. Compared to quantitative resistance in DMIs or group 3, it's erosion of disease control from sensitive moving gradually until to get resistance. That's the need number of changes in the gene, and it's variable between the member and less stable. How did the resistance develop? Basically, if you imagine that your vineyard infected with two type of populations, susceptible and resistance, black and orange, as you can see here, you need to control it with fungicide A, for example. That will kill most of the sensitive. The resistant one will breed and populate it. And then if you keep doing the same fungicide or from the same group, you will end up with dominated resistance population, and the fungicide is not working. That's very, very simple term of doing it. So now we need to look what is not fungicide resistance. So not everything you see in the field or in the vineyard after spraying, you see this is not working, or the pathogen come up, you think it's resistance. Poor coverage is not a resistance if you're not covering very well. The wrong dose of fungicide is not resistance. Wrong concentration factor, wrong timing. If you apply protectant post the infection, of course, that's not resistance. If there is a new unprotected growth, so you spray and there is like a new growth coming and infected, that doesn't mean it's resistant, it's just out of chemical cover. And the wrong fungicide selection. So we have an example in our project. We have tested these few chemical from the same group, group 3. You can see the difference between this group. For example, we have a complete control for Pilanti compared to Biafidan. This is all of them in the same group, and they are completely different efficacy for them in the field. So, sorry, in the greenhouse, we have done this experiment. So meaning if you have non-control disease, it doesn't mean you have resistance. It means you have the efficacy of this fungicide is not equal. And there is a very good practical, very good webinar. I'm advocating for a IWRI webinar now. There is a good webinar by Liz Riley and Barbara Hall about the practical steps in encountering fungicide resistance. It will cover all that in very details. It's very useful. So if you've done all that and you review all that and you think you have fungicide resistance, then you are welcome to fungicide resistance. So what are the research step and outcome we hoping for? We looking for to optimize the high throughput sequencing, high throughput laboratory testing, using fancy machine to be able to detect resistance in so many samples, so that the farmer could send this sample and then we can detect it. We optimize the in-field testing as well. That's Lincoln will talk about it. We develop sampling protocol. What's the best way to take samples? And then, of course, we keep monitoring fungicide resistance using high throughput and using genotyping and phenotyping as well. Hopefully, we can develop a new assay for detecting new resistance marker. So how to monitor powdery and downy mildew and bunch rot? Basically, the grower will send us a sample, either powdery, downy, or bunch rot. Sorry, Lincoln, I'm stepping your feet now, but Lincoln will explain more about bunch rot and botrytis. So this sample will be tested using either phenotyping or genotyping. Phenotyping is basically spraying the leaf, the disc leaf, with different concentration of fungicide, and then that fungicide, if it's the pathogen grow in the field rate, we call it resistance. If it grow in 1 in 10, we call it reduced sensitivity. And if it's not growing, we call it sensitive. Same thing we do, we collect the mycelium or the fungus and we extract the DNA for the genotyping, and then we're looking for a specific mutation in that gene, in a specific gene. And if that happened, that the mutation is there, and then the phenotyping it's growing, that means the phenotyping and genotyping are equal, and that's happy day we can use the genetic analysis for that. However, sometimes not all the time it's reflecting what's happened, so we need to look for different mechanism or different tests. We do this analysis using high throughput qPCR or high throughput sequencing. So what do we test for the phenotyping and genotyping? These are the table we use for powdery mildew. We use group 3, group 5, 7, 11, and 13, and then you can read all these chemicals that we're using. We're looking for mutation called Y36F for group 3, for group 7, H242X, and for group 11, G143A. For downy mildew, we're testing for group 4, 11, and 40, and we're looking for this mutation. This is the result for powdery mildew for phenotyping and genotyping. As you can see here, that's the total number we have tested for each different chemical from 21 until 24. For example, tobas, we tested 19 sample, for digger 13, prosper 17, and so on and so forth. So anything, the blue bar represents sensitivity, the orange bar represents the reduced sensitivity, and then the red bar represents the resistance. So clearly you can see the resistance in a group 3 and group 11. Cabrio, we have 32% of the sample have resistance. For Amistar, we have 60%. For group 13, we have Talendose, 67%, and for Legend, we have 57%. Yes, it is scary. You can see big number and lots of red bars, but remember, this is targeted sample. So it's not a random sample. These samples sent by the grower who knows that they have problem in the spray program. So it's been selected. So it's not, I think, as bad as it shows here because it's a selected sample. When we do the genotype testing, we're looking for this mutation. We find all the samples we send it's have the mutation. However, it's not reflecting in the phenotyping. It's only 11%. That's when we say sometimes the genotyping is not matching the phenotyping. So we need to look for different mechanism for that. For group 7, all the sample was wild type, and that's you could see it's reflecting here. There's no resistance. For group 11, we have almost 60% of that. We detected the mutation, which is responsible for the resistance, and you can see almost 60% of that sample has been detected in phenotype. However, we don't have mutation or genetic analysis for group 13 and group 5 because we don't know the mechanism. For downy mildew, we have identified resistance in 23 sample from 22 to 24. 43% of these samples have resistance to metalaxyl or group 4. 9% of these sample resistance to group 11. And then when we do the genetic analysis, we found exactly 9% of these samples have the mutation. However, for group 40, we don't have any mutation detected, and most of they are sensitive. This is a case study to show you what we get from the grower. Normally we have this sample code and the day they collected, the season, where it's come from. I like you to focus on this highlighted in green. So this isolate has been suspected with DMI, digger, or Talindo resistance. The same thing with this isolate has been sprayed with lots of Talindo in the season for an experiment. The table, I'm not expecting you to understand all that table, but that's how the results look like. For each isolate, we tested these against all these chemicals, and we analyzed the data. So you can see back again to this isolate 332, they have X number of Talindo spray 14 days apart, and guess what? You get resistance in Talindo. And then for these two isolate 318 and 327, they have suspected DMI, and we got resistance in DMI here, and then reduced sensitivity in here. So that's kind of information very useful for us to understand what's the status and what they think for the grower. So how do we collect the sample for genetic analysis? We basically using either a spore trap, it's a simple device on top of the canopy collecting spore from that canopy, or using cotton bud, or we're using a mini vacuum to vacuum all that mycelium, or we're using like a swab from gloves or from your hand after you finish work. This is the field trial of the rotorot spore trap conducted last year and this year from August to February 2024. We sample every 2 weeks, and then we run the spore trap for 2 days. And we thank all these people to participate in this field trial. Basically, we have 2 spore traps in New South Wales, 2 in Victoria, 2 in South Australia, and 2 in Tasmania, and 2 in Western Australia. So they sample every 2 weeks, and they send us this rod, and then we can analyze them. This is the result for this sampling. As you can see here in Margaret River, we have detected the mutation on the mid of October and the end of December. For Barossa Valley, almost all the year around, we can detect the mutation. For limestone, the same story. For Riverina, we got at beginning of October. For Hilltop, we got almost the end of the season. For King Valley, mid-November and almost the end of the season. For Yarra Valley, we got end of December to early January. For Southeast Tasmania, we got early January to mid-February, and for Tamar Valley, we haven't detected any mutation, which is good. So which collection method we could use? Basically, you could use any collection method. It's all working. So you could use a cotton bud, swab your gloves, or mini vacuum. The grower prefer these 2 methods because they are easy and practical. So the conclusion would be we have developed a marker for group 11. We call it G143I, QI, EN. This is proposed name for it. So we can use it for high throughput qPCR, and then we can sequence a lot of samples at the same time. For the rotor rod and qPCR is very effective. So it's the grower can send their sample, and then they would know what's going on in their vineyard. All samples are equally effective. Sample methods are equally effective, and you can choose whatever you like to more practical. We have identified powdery mildew in group 3, 11, 13. For downy group 4 and 11, we have found resistance. For Y360F mutation in DMIs, it's not linked to the phenotypic resistance, and we're still working on that. So hopefully there's more resistance to come. So the take-home message of that work, we can test what we have in your vineyard. We can know what's the population you have, and that's help you to make a decision on what you can supply in the next season or the next supply. I'm going to hand this talk to Lincoln Harper from Curtin University. He will be talking about botrytis and sample submission. Thank you so much.
Thank you, Ismail. Just like to remind people at the other end, if they've got a question, feel free to type it in and send it through, and we'll hold a question and answer session after Lincoln's presentation. So our next presenter, our next speaker this morning is Lincoln Harper. Lincoln Harper is a research officer within the Centre for Crop and Disease Management at Curtin University in Western Australia. Lincoln is currently working within a CRC SAAFE funded project, which is characterising fungicide resistant in grapevine fungal pathogens. His research is focused on botrytis in wine grapes and is also involved in the genetic work in grape powdery mildew and grape downy mildew. This research aims to provide valuable information that could contribute to improving resistance management strategies in an economically important crop. So Lincoln, if I just, sharing screen, great. I'll just hand over to you.
Awesome, thanks for that, Matt. And yeah, thanks for organising the webinar today. And thank you for everyone tuning in today. Lincoln Harper here from Curtin University. So I'm just going to cover the botrytis work today. And yeah, Ismail's already covered the mildews, and also I'll finish up with talking about some of the take-home messages from Ismail and I, and also just explain how we things around sampling and stuff. So for botrytis cinerea, so what do we basically test for? So for botrytis, we're interested in 3 main groups. So that's your group 9, group 12, and group 17. And so those are, there's the active ingredients in those groups are in your Scala, your Switch, and your Taldor. I've also done some testing with group 7 and group 11. So that's your Phalan and Amistar. So the focus is on the 9, 12, and 17, with sort of less of a focus on the 7 and 11. And for each of those groups, there's at least one gene that we look at for mutations. For group 9 and group 7, there's actually more than one gene. So just quickly on the method, it's very simple. So basically, the majority of the time I receive swabs from a grower. They've basically swabbed expressing botrytis late in the season from their vineyard. Then I've cultured that botrytis from those swabs, and basically I end up testing isolates or strains of the botrytis on 1 dose of fungicide. So basically it gives me a yes or no. So I've worked out a dose of each of these compounds that will give me a yes or no that's resistant or sensitive. And once it's resistant, then I look at the genotyping, which is basically looking at these genes. So amplify these genes and look for mutations. Okay, so just the result. I've just got the last 3 seasons, so 2022 to 2024. So the last 3 seasons, I looked at around 370 isolates across 43 vineyards, and that's across WA, South Australia, Victoria, New South Wales, and ACT. So this column graph is basically on the vertical axis. I've got frequency, and on the horizontal, I've got different chemical groups. So those 3 main groups, the 9, 12, and 17. So I've just broken the data here up into a per chemical, sort of like a per chemical basis. So each column represents frequency just for that chemical. So it's not considering any isolates that are resistant to more than one of these chemicals. So basically for group 9, you're looking at around 15% frequency of resistance, and for 12 and 17, you're looking at around 10. So that's the red component of the column, and the blue is the sensitive. The pie graphs are showing just showing some of the genotyping results. So for group 9, there's a fair amount of different genotypes we can find spread across 2 genes. For group 17, there's sort of a handful of changes, but they're all in 1 gene. But basically, take-home message here is for each of these 2 groups, group 9 and 17, there's usually a predominant change. So for group 9, that's L412F, and group 17 is F412F, or F412S. So the majority of these isolates resistant to each of these groups usually hit that mutation. So I've just gone ahead and broken up some of those phenotypic results. Broken them up into phenotypes basically. So when I mean phenotype here, so basically if these isolates are resistant to either one or more than one fungicides, I've called that a phenotype. So in the top left here, you've got basically a legend with the different numbers represent the different chemical groups. So you've got basically 7 phenotypes here. So basically different combinations, or just basically one phenotype can just be resistance to one fungicide group. So for the heavily sampled regions or states, WA, New South Wales, and Victoria, got these large pie graphs, and it just breaks up all the phenotypes. And I guess again the takeaway here is majority of the isolates are usually either resistant just to group 9 or just to group 12, or a combination of the 2. So that's your sort of light blue, dark blue, and orange parts of the pie graph. So most of them are, a fair chunk of these isolates are just resistant to one chemical group, and there is a few that are resistant to more than one. South Australia, we didn't have any resistant isolates. ACT, we had a handful of isolates resistant, and they were just resistant to group 9. And just the overall frequency of the whole, so you're looking at the whole population for the last 3 years. If you look at the overall frequency, so different from the previous slide where I was looking at frequency per chemical group, if you just look at the overall, so any kind of resistance, whatever form, you're looking at around about a quarter of the isolates are resistant. Okay, so just the spore trap results for Botrytis. So Ismail just touched on this. So we're basically running these spore traps essentially most of the season. So for Botrytis, we were looking at group 9 target, L412F. So each column graph is showing you the results across some of these different regions. So any bar, any column that exists basically means there's detection. If there's no column, there's no detection. And the red indicates resistance, and blue is wild type, sensitive. So basically across the regions, generally there was a fair amount of detection across the whole season, and it's obviously a little bit patchy. And some of these regions were reasonably well sampled. So basically every 2 weeks we're having different rods come out for testing. And also the resistance bar popped up as well in some of these regions, and this obviously you might notice there's also mixtures, so we got mixed columns there. But basically, this method, we're basically looking at it as like an early detection system. So looking at something we can look at detecting Botrytis early in the season, or mildew, because a lot of the sampling, like I said, I do at the moment is basically from sampling at the end of the season. So I'm getting swabs from expressing Botrytis. But this system potentially looking at basically detecting resistance early in the season, so providing some early indication of resistance in a vineyard. So another thing we've been working on is basically an in-field qPCR system. So basically we can test for the resistance in the field or in a vineyard on the day. So we have a small mobile qPCR machine. So we can basically on site we can extract DNA from infected material and run analysis and look for resistance genotypes. So it's a very simple system, small machine. It's fairly affordable, and it's sort of happening over 2 or 3 hours basically once you get all your sampling done and do your testing. So just some of the pictures here are just showing basically Botrytis infected berries. Basically hand grind them, and then you dilute the slurry out, and then run it in a small qPCR machine. And on the far right, you can, in this case, we just kind of mapped out where we were sampling in that little block and showing basically hits of yes or no, basically if the mutation's there or not. So it's at the moment, it's a nice like, first look. So you say that you've got a problem, you think you've got a problem. It's a nice way to go in and have a quick look at a vineyard. Because there is limits, obviously. It's a small machine, so there's a limit on the sample number, and also you can only do so many mutations at once. So I think it's something it's a quick look, first look, and then you'd follow that up with more thorough testing. So you'll go back and take some more samples and go back into the lab. So sort of that preliminary look, really, basically using the system. But it's really sweet the way it is, the mobility of it, and fairly obviously very rapid, and fairly cheap. So just some of the conclusions from the Botrytis stuff. So just basically the lab testing of the field isolates that I've been talking about. Yeah, frequencies are fairly low on a per chemical basis, so you're around 10 to 15%, and the overall frequency is 24%. So with most resistant isolates are resistant to only one chemical group. So if you looked at those results, you're basically saying you could look at some of these isolates you're getting from a vineyard. Most of them are only resistant to one chemical group. You can use another chemical group to control because they're only resistant to one. There is sampling bias here, so as Mel touched on that, obviously sample size. You might get 50 swabs from one place, you might only get 5. So there is issues with how many you get, and also blocks. Probably a fair chunk of the time you get samples from blocks that have previous issues or suspected issues. And also there is a small group of vineyards that are now popping up with high frequencies. So I think I'm really keeping an eye on some of these. So generally most vineyards are great, but some vineyards are popping up with fairly high frequencies. So they've obviously had a fair amount of use of fungicides over the years. So basically, moving forward, we want to keep maintaining monitoring of these critical mode of action. So that's your 9, 12, and 17. So there will still be those phenotyping and genotyping techniques. So sort of the backbone of the project. So basically, probably still a mix of lab and in-field work. We still want to keep monitor. We basically, ultimately, we want to monitor each season. And in terms of what goes back to the grower, you'll have some results with basically a phenotype, so yes or no to a resistance to a certain chemical, and also the genotype. So that's provided back to the grower in a timely manner, hopefully before they start working on their next chemical program for the following season. So the spore trap stuff, it's going to be a useful tool for early detection, especially when you can do something early in the season, so in spring. And with the basic conclusions from those results, Botrytis was detected at most sites. Pretty much all of them. Again, detection, you can do it basically all or most of the season. So we're going to repeat that again. So we want another data set basically on top of what we've already done. And also, I think as Mel touched on that, we can also add other targets. So I guess for Botrytis, that was just one target, one chemical group. From there, you can always add another group. So I've got the other group 17. I've got another assay for a different target for that group. Okay, so just some of the take-home messages here, basically around resistance management. So ultimately, a lot of the work out of the project is providing information so that we can then go back to a grower and we'll give them information that will help them adjust or improve resistance management strategies. Basically there's a lot of monitoring still, so we want to still keep monitoring basically in this project. Some of the rules here, you might say, rotation. So rotation's a big thing here, mixing different modes of action, using the label rate, and limiting the total number of applications per fungicide group. So a lot of these themes here in crop life and the dog book, basically just understanding what you're applying. So educate yourself about the type of group you're using. So obviously there's lots of different formulations out there, but they might, some of them are the same group, different formulation. And basically the last one here, start a fungicide program with a multi-site mode of action to reduce populations. So do not use a single site mode of action to control well-established infections. So just some take-home messages here around resistance management. Okay, so the sampling. So we always want more samples. So it's free. So Ismail's in charge of dealing with the mildew, so the downy and powdery samples, and I deal with the Botrytis samples. So we're always interested in new samples. It's free, and like I said, we all like to. It's a win-win for us. We need to test samples, and then we like to give win-win for all of us, and we like to give those results back to you guys in a timely manner. So basically, we've got a couple of. This is just showing with a couple of. Basically, we've got a sampling sort of protocol for each of the pathogens. We also have a sort of a document that allows you to fill in stuff about spray history and stuff. So we've got a bunch of documents for the sampling. So if you're interested, yeah, feel free to email Ismail or myself using the emails here in this slide, and we can provide you with these documents. And we can talk to you on the phone or whatever and run through the sampling and whatever. So yeah, just we're really keen, still keen for samples. So we can send these things out for you, and yeah, we can go ahead with it. And yeah, this is just my acknowledgement slides. So just basically the industry reference group and all our collaborators, and a big thanks to our funding bodies, Wine Australia and CRC Safe. And thank you.
Thanks, Lincoln. That was great. Just like to remind people, if they would like to send a question through, just type it into the Q&A and press enter, and it will come through. And both Ismail and Lincoln are here to answer any questions. Lincoln, just with the sampling protocol, how many times a season would someone be likely to sample? Is it just the once?
Oh, I guess my experience with Botrytis, it's probably usually just the once, right at the end of the season. But I mean, it's not an issue to get both. I mean, I'm happy to get some once latent infection just starts expressing at the start of the end of the season, and also get some so many weeks later if they're interested in looking at that, because frequencies could be changing across that season, the end of the season. So from when you first get expression or when you get if you're getting full-blown disease in your vineyard. So generally once, but it's not a big issue to do it more than once. So yeah. I know Ismail might comment on mildew, how we're sampling mildew.
Oh, just pop this.
Yeah.
You're just on mute there, Ismail.
Yeah, thank you. So yeah, mildew and powdery and downy is not that easy as botrytis, as you know, because it's obligate bioassay. We have to test it on actual leaf. But if you need sequencing for a group 11, that's definitely possible. You can send as many as you like for a group 11. For other group, yeah, possibly we're looking for sample once a season. That's fine. And if you're really keen, you have a spray program and you want to monitor your population, that's also possible. Yeah.
Okay, there's been a couple of questions come through. The first one is, what trends have been observed for, I think that meant, 9 and 17 over recent years?
Yep, yeah, I can answer that. So yeah, basically, I mean, I've been working with Botrytis for these two groups for quite a while now, probably 8, 9, 10 years. And basically group 9 and group 12, they're fairly consistent in terms of frequencies across Australia. So they're fairly consistent around that 5 to 10% mark. So they haven't changed overall. They haven't changed a lot. But I think what I was saying before, there is the odd hotspot. You might call it a hotspot vineyard, where frequencies are fairly elevated. So I think overall, I think there hasn't been much change in the trend, but I think I'm noticing that there's the odd hotspot vineyard popping up here and there. So it's something we need to keep an eye on. So I think generally it's okay, but I think there's still some hotspot places we need to really keep an eye on and work out what's going on there.
Okay. There's I think it's a technical question for you here, Lincoln. After running a qPCR, how exactly do you know that this grapevine is resistant to fungus or not?
Grapevine is resistant. Oh, so I'm looking at the qPCR is looking at finding a mutation that basically is associated with resistance in the fungus. So you're talking about sorry, I'm a bit confused about the question.
Yeah, I think he meant grapevine is resistance to the fungus, which is not that's
yeah
about the resistance for the fungus to the fungicide, the other way around.
Yeah, sorry, is that what you're asking?
Sorry, can you
Yeah.
see the question there, Lincoln, in your?
Yeah, I can.
Yep,
I
yep,
think
yeah.
I can see the question. I'm a bit confused if it's
Okay, maybe if the person who's asked it, maybe just elaborate a little bit further. There is another
Well,
one.
I think it's mixed up between grapevine resistance and pathogen resistance. What you're asking, my understanding, what you're asking for is that how do you know that qPCR it's grapevine resistant? It's actually not we're not sequencing for the grapevine. We're
yes,
sequencing for the pathogen to know if it's the mutation in the pathogen make it resistant to the fungicide. We are not looking for the grapevine, is it resistance to the pathogen or not? If you understand it. Yeah.
Yep, there's a thumb up, I think. There's another question here. It says, with some of those resistances noted for groups 9 and 12 for Botrytis, or 3 and 13 for Powdery, will it get to the point in the near future that those groups will become ineffective?
Yeah. If I talk about Botrytis for 9 and 12, yeah. I mean, it's possible, but I think at this stage, the way I've looked at it, where I've seen it over the last few years, I think that, it's possible, but I don't think it's in the near future. I think I've seen most of the management seems really good. So I said these frequencies are fairly low, like 10 or 15% is great, really, compared to, well, if you compare it to overseas, some of these other reports overseas are far higher. A lot of them are far higher than us. So I feel like management's pretty good, I think, for Botrytis. So I think it's possible, but I don't think in the near future.
Yeah. For group 3 and 13, theoretically, yes, will be ineffective. If you keep doing spraying the same group over and over again, eventually you will actually breeding up for that resistance, and that your population will be dominated by the resistance. There is an example in Europe. They lose one fungicide in 2 years because that fungicide was too good to spray and very controlling, and they keep spraying it until they have to draw it from the spray program. So yes, it can be ineffective if you abuse it.
Follow-up question, I guess, relating to the chemistry and becoming resistant. Is there any new MOA chemistry coming to add to the rotation? So I think that.
Yeah. From what I think there is new stuff coming, but I'm not sure if it is going to be a blanket release or blanket registration. So I feel like there's new stuff coming, but I don't know if it's how widespread it's going to be. I don't know if there's Malcolm comment on that. For
Botrytis and Powdery, I got notified that there is a new chemical called Verbixol. It's from group 21.
Oh, 21. Okay. Yeah.
From the market.
So it's coming. Yeah.
To Powdery and Botrytis. Yes.
Okay, so there's one coming. Yeah.
But the 21 is still it's already in the list, but that's a new chemical. Yes.
Okay, there hasn't been any other questions come through. Just gonna. Okay, what we might do is we might leave it there. So look, thank you again, both Ismail and Lincoln, for this morning, or for today, for providing a great insight into fungicide-resistant Australian vineyards and also some strategies. Just like to remind everyone that they will receive a link to this recording, and which will be uploaded to the YouTube channel in the next 24 hours. We will also provide some linkages towards both Lincoln and Ismail to submit samples in that email that will be sent out as well. And if you have any questions, feel free to get in touch with either Lincoln, Ismail, or the Adabri, and we'll be able to put you in touch. Thank you again today, and I look forward to seeing you at the next Adabri webinar. So keep a lookout on the Adabri website, and we'll be advertising that in the coming weeks. Thanks again.
Thank you.
Everyone. Thank you.